Polymerase chain reaction, also known as PCR, is a molecular technique that amplifies specific DNA or RNA sequences, allowing for detection and quantification of genetic material that's even in very small amounts. So for the principle, PCR uses thermal cycling to repeatedly denature DNA, anneal primers, and extend new DNA strands. This process exponentially amplifies the target genetic sequence, enabling sensitive and specific detection. So how it works is we have denaturation that occurs at 94 to 98 degrees Celsius. So that's where the double-stranded DNA is separated into single strands. Then we have the annealing, which happens at 50 to 65 degrees Celsius, where the primers bind or anneal to the specific target sequences on the DNA. Then we have the extension, which happens at 72 degrees Celsius. The DNA polymerase synthesizes new DNA strands starting from the primers. Then we have thermal cycling, so the cycle repeats 20 to 40 times, amplifying the target region exponentially. PCR can be used for pathogen detection, so it can identify bacteria, viruses, and fungi by detecting their genetic material. Genetic mutation analysis, so this detects inherited mutations, oncogenes, or chromosomal translocations. It can be used for gene expression studies, so it can measure mRNA levels after converting to cDNA to evaluate gene activity. It will also be used for forensic science and paternity testing, so it can amplify DNA from minute samples for identity testing, even with degraded or limited material. Real-time PCR, so this quantifies DNA or RNA in real time, which is useful for viral load monitoring, gene expression, and diagnostic thresholds. Some advantages is it is highly sensitive and specific, so it can detect low copy numbers. It will give rapid results, often within hours. It is versatile, so it's applicable to DNA or RNA or multiple organisms and genes. It also has quantitative capability with real-time PCR. Some things to remember for PCR is PCR amplifies DNA or RNA through thermal cycling, allowing for detection, quantification, and mutation analysis. It is essential in clinical diagnostics, infectious disease testing, genetics, and forensic science. Real-time PCR enables monitoring of DNA, RNA amplification as it happens, providing both qualitative and quantitative data.